we need to talk about genes. We're going to get started with genetics, and this is kind of an introduction to how genes get from one place to another. We haven't defined genetics. We haven't even really defined genes, but I think that most of you probably already know what these are. I hope so, anyway. We have to start with chromosomes, and we talked about this with mitosis, that chromosomes contain DNA, and genes are made of DNA. We're getting a little bit ahead of ourselves, but that's okay. All cells have their genes contained in packages called chromosomes. And here the arrow is pointing to a set of chromosomes. A full set of chromosomes consists of 2 copies of each one. 2 copies of each one. And we use the word, there's a word for that we use in biology that's called the diploid number. Diploid number. Di means 2. So, if you look at this set of chromosomes here, you can find a pair for each one. Okay, there are 2 copies of each chromosome here. Here's an easy one to find right there. Okay, because they put the red dots on them. I don't know why the red dots are on these. This is just an image I downloaded from the internet. So if we look through these sets of chromosomes, in fact, here's another pair. Okay, and you can go through those, and if you're bored while you're watching this, pause it and go through and find all the pairs. There are 46 chromosomes here. 46, because this is a human set of human chromosomes. Humans have 46 chromosomes and 23 pairs. One of these pairs came from your mom. One of these pairs came from your dad. Okay? They use the word maternal chromosome and paternal chromosome for mom and dad. Mama and Papa. Okay, so you can do that for all those chromosomes there. Other organisms. Here are some sample chromosome numbers. And I should have prefaced this probably by asking you to think about how many chromosomes do you think other things would have. And it turns out that the size of the organism is not really indicative, doesn't really indicate how many chromosomes something has. For example, a fruit fly has 8 chromosomes, a dolphin 44, a cat 16, but a chicken has 74, even though a chicken is smaller than us. Their chromosomes are just littler. There are certain organisms, like certain plants, that have tons of chromosomes. One thing you'll notice about all these numbers is that they're even. Okay? Why are they even? Well, because an ant, for example, has 2 chromosomes. It gets 1 from mom and 1 from dad. A cat gets 8 from mom and 8 from dad, and so on. So, the number of chromosomes an organism has will be even. Term karyotype. A karyotype is a picture of all of the chromosomes of an organism, and they've been lined up. To do a karyotype, you take all those chromosomes. If I go back to that slide we just showed you a minute ago, we would take all these and we cut them out with we literally take a piece of paper and cut them out and paste them together with their partners. Your chromosomes are numbered by size and where this little thing in the middle is. That little thing is called the centromere. And so you'll notice that it generally goes from big to small, where chromosome pair number 22 is really small. Shown is a normal blank human karyotype. Male or female is the answer there. And the answer to this one is male. How do you know? Well, if you look down here at the last set of chromosomes, Males, if you do a karyotype, will end up with two chromosomes that don't look alike. We call those chromosomes the X chromosome and the Y chromosome. Because literally, if you see this chromosome, it looks like a little upside down Y with a little tail on it. And that one, if you see it, literally it looks kind of like an X. So they call it the X chromosome and the Y chromosome. That's a male. We use a term for these chromosomes. We use the word homologous. Homologous or paired chromosomes are the same length and they have the same banding pattern on them. And again, humans have, I don't know why I've repeated that statement, but they have 46 and 23 pairs. Here's another pair, here's another set. Okay, this one looks very much the same as the last one. You can't see the banding pattern very well, except the last pair look the same. That would be two X chromosomes. That would tell you have a female. It's a female karyotype. You know it's normal because every chromosome is paired up. In lab, we'll look at a couple examples of non-alike chromosomes. So how do you get them? You get them by a process we're going to talk about. We said that you get a copy of your chromosomes, one from each parent. Your parents pass them on to you in their gametes. That would be your mom passed them on to you in her egg, dad in his sperm. Your dog got an egg from its mother and a sperm from its father. And those gametes contain one copy of each chromosome. One copy of each chromosome, and we use the term for that as haploid, half the normal number. In humans, that would be 23 chromosomes in the egg and 23 in the sperm. And when egg and sperm get together, 23 plus 23 is 46, or your normal number. To make gametes, our bodies do something called meiosis, or if it helps you keep it different from mitosis, meiosis. Meiosis is the process of making egg and sperm, or pollen and ovule, or haploid cells. So we're going to spend a few minutes talking about that process. Meiosis has the same phases as mitosis, just two of each. Now, if you think about this for a minute, to do mitosis, you started out with a cell that had 46 chromosomes, and you doubled them to a cell that has 92, because you made a copy of each chromosome. And then that cell divided into two cells, each that had 46 chromosomes. We're using humans as an example. Meiosis works the exact same way, except there's another division. So that each one of these cells will have 23. There's a whole other division. And I've listed here for you the phase names, and it's not that important. They just add ones after the names. Okay, so stage one of meiosis is a little bit different than stage one of mitosis. In meiosis, in prophase, chromosomes find their homologous partner and pair up, forming tetrads. What does that mean? Well, that means chromosome number 1 is going to find the one that looks just like it and get right next to it. And chromosome number 2, let's do chromosome number 23 because it's really, or 22 because it's really small. Chromosome number 22 will pair up with its little partner, and so on for 23 pairs. They form what are called tetrads because there are 4 chromosomes in each pair. Sometimes, when they do this, they will cross over. They'll overlap each other, and this chromosome will end up with a piece from that chromosome, which makes their genes a little different. We'll talk about that a little bit later. Okay, and if you were to go, you can go to this web page that I just hyperlinked on your notes, and view it. I'm not sure it will show up very well if I do it on the video. So in metaphase 1 to telophase 1, homologous chromosomes go together to each side of the cell. So in other words, our chromosome pair number 1 here will split, and you'll end up with 2 of those. Instead of in meiosis, these X's got pulled apart. So when we're done, we have 2 cells in humans, each with 46 chromosomes. Then comes a phase called interkinesis, which means between divisions, where there is no duplication of chromosomes. Then comes the second stage. Prophase 2 to cytokinesis proceeds exactly as mitosis, but as I said before, you get 4 haploid daughter cells. They have half normal number of chromosomes. They'll have 23 each. And again, refer to those animations to help you understand this, and we'll talk about this more in class. When we look at karyotypes, we're going to see some problems. And one of those, the reason for those problems is something called nondisjunction, which I think is a really cool word. It's a double negative. Non means not, dis means not, junction means to join. A disjunction is when you separate. Literally, this whole word means to separate. And this means to not separate. Obviously, or maybe not so obvious. In non-disjunction is when homologous chromosomes do not separate themselves during meiosis. So in other words, here we have chromosome pair number 1, let's say, and chromosome pair number 2. In the first part of meiosis, this one goes that way and this one goes that way like normal, but you see here that these two did not. So after meiosis 2, when they divide, you end up with, this cell ends up with 2 chromosome number 2s and 0 over here. And so this cell here ends up with an extra chromosome, and this cell ends up with a missing chromosome, written as n-1 and n+1. The result is a cell with too many or too few chromosomes. If this is a sperm, if these become sperm cells, I'm drawing little tails on them, and this sperm fertilizes an egg, that means that, okay, this sperm has actually 1 more chromosome than it should. It has 24. When that fertilizes an egg that has 23 chromosomes, that adds up to 47. 47 chromosomes is bad news. If this sperm fertilizes an egg, that sperm has 22 chromosomes. Meets an egg with 23 chromosomes, that's 45 chromosomes, bad news. Here are some genetic disorders caused by this problem. Down syndrome is when you actually get 3 chromosome number 21s. Some of you here know people with Down syndrome. Okay? Characteristic traits of Down syndrome: high forehead, flat nose, almond-shaped eyes, no philtrum, really, flat upper lip, tiny upper lip, very happy usually people, not usually mentally capable of getting much beyond an elementary level, usually. Okay? Down syndrome is very common. In Down syndrome, you get 3 chromosome number 21s. We're going to look at this in lab. There's a disease called Turner syndrome where a person gets an X chromosome from either its mom or its dad and nothing else. It doesn't get either an X or a Y. There's a disease called Klinefelter syndrome in which a person gets an extra X chromosome from its mom or an X and a Y from its dad. And this person is male, but they'll have problems. This person is female to look at, but they'll have problems. They won't be able to have kids, they'll be sterile, and there are other issues with that. There are no real other diseases that you can live longer than a year or two with extra or missing chromosomes. Most babies that have extra or missing chromosomes, most, are spontaneously aborted. Which literally means a miscarriage. If your mom or your sister or your aunt had a miscarriage, they think that many times the baby's had something too many or too few chromosomes.